We are going to hear from Muzlifah Haniffa. She is one of the 3 coordinators of the Human Developmental Biological Network. So she will give us some insights on that too. She's a dermatologist with a research background in immunology and has pioneered the application of single-cell genomics to study human development. The main focus so far has been on decoding the developing human immune system. She will talk today about the Developmental Cell Atlas. Welcome, Mus.
Hello, everyone. I hope you can hear me and that I'm on full screen slide mode. Thank you very much, Patricia, for the organization. It's very exciting to be on this meeting today, the first for the Human Cell Atlas Latin America. And I'm going to be talking about the Developmental Cell Atlas. And I speak on behalf of the Developmental Biological Network, and as one of the 3 coordinators who include Deanne Taylor and Stanley Nassen. And then if at any point you would like to get in touch with us to join and participate in the Developmental Cell Atlas, please do not hesitate to email development@humancellatlas.org. Now, human development is quite complex to chart, partly because cells are changing in composition, in type, and also in morphology, and there is the added layer of time, whereby gestation in human up to 9 months, where things are extremely dynamic. And much of the effort going into studying human development has to be done by taking serial analysis, almost snapshots, and then putting that together to have the kind of idea of what happens over the 9 months gestation. And also by profiling either specific organs or anatomical units from the embryos or fetuses. So really, the Developmental Cell Atlas has to take into account of the anatomical space and gestational time, which both affects the individual organs and the whole embryo or fetus. So the first thing to do is to consider the ethics around the Developmental Cell Atlas, and there are many regional regulations which are very different across North America and Europe. Also the availability of tissue banks and repositories or bioresources to study human development. In Europe, at least in the UK, we have the Wellcome and MRC funded Human Developmental Biology Resource, which is dedicated to provide samples to study human development between 4 to 20 weeks. And in France, there's the recent establishment of the Human Developmental Cell Atlas funded by INSERM, which also provides a resource, including pre-implantation material for studies. Now, how do we build a developmental cell atlas? I think the talks earlier have alluded to this and makes it easier for me to explain. Well, firstly, we need a cross-disciplinary team. We need individuals who are from the biological community, medicine, computational scientists, and also technologists, engineers, chemists, and physicists. Because there's a lot of using cutting edge development, being able to extract the biology, being able to kind of cope with the huge amounts of data and making sense of it. The second is the different modalities that you can use. We have the imaging platform. I mean, really, for many years now, centuries, in fact, we've been looking at tissues and trying to kind of look at the individual units that form a cell, tissue, and so on and so forth. And now we can measure many, many messenger RNA within single cells, the regulatory regions, the DNA, and the protein. And then we want to be able to go beyond looking at cells that have been dissociated from tissue to looking at where all of these RNA, protein, DNA are in the tissue context in situ, spatial level. And then we have all of these information which we then have to integrate using computational tools and algorithms. And the measurements really ranges in a sort of a massive scale of anatomical scale, tissue histological scale, cellular and molecular scale. And all of this then needs to be integrated and then made coherent to kind of like build the atlas. And in terms of the atlas, any atlas, but particularly the developmental atlas, one can easily imagine the sort of like tangible output of this atlas, i.e. a sort of a reconstruction of what a whole embryo is, or what an organ within an embryo is at particular gestational stages and across time. But there's also the kind of intangible information and data. And these are kind of like patterns that one can abstract from that information that will tell us about the biological system that may or may not be apparent to us from just looking at the individual components of the data. What we ultimately want to do is to understand human development. What are the cell and molecular programs that underpin development, how that changes across gestation, and how it's in relevance to disease. So there's kind of trying to understand all of the cells within the X, Y, Z coordinate, and also the temporal component, trying to basically understand what cells are, where they are, and then what are the variations that can happen in terms of the environmental impact and also the genetic background, and what basically allows the cells to develop in a particular line of direction, the fate decisions it makes, and how this becomes really this canalization of how, you know, we all kind of like develop organs in particular manner. So all of these challenges, the technology, the computational challenges, and also the having a coordinate framework, which can be either absolute, i.e. like a zip code of where a cell is supposed to be, or relative, i.e. a positional landmark in relation to an important anatomical site. These are all things that we have to really develop ourselves. It's quite challenging. If there is no way you could create one, but just imagine where we can go, what we can unearth and learn from the Developmental Cell Atlas initiative. We have to bear in mind that we will never be able to get all of the stages of human development just because of the inaccessibility of material and also some of the kind of difficulties associated with the ethics, etc. And here we can rely on some of the advances that have been made in developmental biology, the model systems of early human development, where you've got, you know, gastrulation in vitro models, and also, post-gastrulation models. In addition to these in vitro models, we've also got the organoid systems, cell cultures, and also animal models, which are extremely attractive for at least one other reason, in that you can actually perturb these model systems in vivo. It's much easier to do, you know, fate mapping studies, and to actually understand, to kind of like delve into the mechanistic insights. So we can use the human reference data, have data-driven hypotheses, and then dissect specific features in animal model systems, but also perform comparative biology analysis, so you can actually understand what are the features that are conserved between the species, and which are factors that are quite unique to human and in contrast to other model systems. And within HCA, we have other biological networks, and we've got the organoid network led by Barbara Treutlein and Christoph Bock. And here is where the developmental biological network can interface in a seamless way to really make inroads into human development. We've made significant progress. There are many developmental atlas publications so far. These are all of the organ-based atlases that have been generated, many of which we've contributed to from within the UK initiative, and also publications that show how the developmental atlas actually leverages suspension of tissue-dissociated measurements and incorporating spatial technologies to kind of like allow you to have a more comprehensive profile of the organs during development. And these are manuscripts from the Swedish effort on the heart, and also in the developing brain. The UK Wellcome funded initiative has focused more on the developing human immune system. And this is quite complex because the blood and immune cells are made in different organs, beginning from the yolk sac, AGM, liver, and the bone marrow, then they subsequently go to other tissues where some of those cells differentiate and then mature, such as the thymus for the T cells and spleen for the B cells, and then also the peripheral organs, where these cells are then subsequently nurtured in their new environment, providing the immune network for the peripheral tissues. Again, here, just to illustrate the complexity of space and time that needs to be accounted for when studying human development. But we've made fundamental insights on prenatal immunity. We've looked at how the intrinsic properties of the hematopoietic stem cells in terms of the progenies that it can produce, how it changes from its location in the fetal liver across gestation and also across the anatomical tissues where HSCs can reside. And this tells you that also the extrinsic environment or extrinsic factors within the microenvironmental niche that then modulate how the immune network is generated and designed. And also how you have the adaptations of the immune cells in the peripheral tissues where they subsequently are nurtured and adapt to kind of encounter antigens in the periphery. What we would ultimately like to do, and we are in a good stage to do this, is to move towards generating a whole embryo atlas. And here we have to reconcile several important factors and challenges. One is, how do we leverage the kind of like macroscopic scale of whole embryo analysis? And that's been significant inroads made by looking at tissue clearing and light sheet microscopy imaging by Alain Chadertal in Paris, and also kind of like reconciling that with the very detailed and high resolution profiling of lots and lots of cells from different organs. And perhaps one way of going about it is to dissect out anatomical units and perform tissue dissociation and spatial analysis of a whole embryo to reconstruct this atlas. What will it do? It will tell us also the emergent properties of the whole human development in toto, where the whole is greater than the sum of its parts. Things that, you know, one way of looking at it are the kind of migratory bird patterns, which you would never observe if you were to study the bird on its own. But it's only when the bird is in a flock with others that you start seeing this pattern. And a bit like if you were on the 3rd floor of a building compared to the view from the 30th floor of a building. But ultimately, this is very much about how we can have a reference atlas with clinical relevance and applications. Lifespan and aging being very important, how we can actually use the atlas to understand developmental disorders and childhood diseases, such as cancer, which may have their origin in utero, and also the application of the reference atlas to innovate tissue engineering and regenerative medicine. So one of the publications to show is the development of the T cell and their differentiation in the thymus and also across lifespan. And just to show that the data can also be accessed at the web portal there. And when we looked at this, it's not just about the T cells alone, but the T cells in their environment. It allows us to look at the cell-cell interactions there, how the TCR, BDJ recombination occurs. And again, looking at what's specific to human T cell development and how it contrasts with mouse T cell development. And I mentioned about understanding childhood cancer or developmental disorder. And this is one example by Sambajati looking at the Wilms tumor, a very common kidney childhood cancer. And the transcriptome of the Wilms tumor shows that it's likely to be aberrant nephron development. And the finding is significant because instead of cytoreductive chemotherapeutic approaches, one can now try to innovate therapies which basically allows the aberrant nephron development to continue and become a mature cells instead of cancer cells. And by extending the developmental cell atlas reference data to the pediatric cell atlas, which is another initiative within HCA, one can really start to understand how things change prenatally and in postnatal life. And just one example of how we can use the developmental data sets for tissue engineering is work that in collaboration with Carl Kohler and Sarah Teichmann, where we've taken the fetal skin data sets from 5 to 20 post-conception weeks and compared that with the in vitro derived IPS derived skin organoid. And you can see how immune cells do not feature in the organoid And we can now look at what are the secreted factors and cell-cell interactions that occur in vivo that may not be apparent or occurring in the organoid model to enhance tissue engineering and regenerative medicine. Ultimately, the data we want to share and make accessible to everyone. This needs to take into account of data protection regulation. And we've heard earlier from Enrique of how we can use repositories, including the HCA data coordination portal to disseminate the data. Another way of data dissemination to a broader community will be through web portals. There are many of those. Here's one which hosts the developmental data sets, and where you can actually browse the web portal to look at what cell states that have been identified, the expression of specific genes, and also looking at the expression by the cell states that you have identified, and also by the cells according to differentiation trajectories and the cell types. So one of the things I wanted to mention about the web portals is how we now need to kind of like move further with basically including genetic disease databases with the web portal, which we've now done for some of the data sets, so that it has a greater utility for researchers, clinicians, and pharmaceutical partners to try and look so that we can actually look at what genes are expressed in which cells that are disease caused, that are mutated in diseases, and also to include the spatial information within these web portals. We're very keen to sort of form a global community, really emphasizing networking and outreach. And we wanted to have our own physical meeting, but COVID-19 came and we couldn't have the meeting. But as always, we've adapted, and actually this has been a fantastic opportunity to start a developmental seminar series. We've had at least 3 of those, and the next one will be on neurons. The dates there, 27th of October. Speakers being Freda Miller and Stanley Narsson. And by starting this seminar series, we've also got one where we have the same seminar, but on a different day and a different time that better suits the Asia and Australian researchers. And the one for Neuron, for example, will be on the 5th of November. So I want to finish here and to say, please do get in touch with us. This is the email link to contact and interact with the Developmental Biological Network. Many people to thank. These are all the individuals who have contributed a lot to the Developmental Biological Network, the HC Executive Office, and also the many funders who have contributed to the Human Cell Atlas Developmental Initiative. Thank you very much.
Thank you so much. This was one amazing talk. So much information that has been gathered already. I suppose one of the questions here has to do with your interface with the organoid networks, considering the difficulties in tissue procurement for your specific kind of biological network, right? The developmental one. How do you, maybe a few words, how are you really seeing the development of the organoids, and how are they, what's the interface that you're building? How far are you on that?
So one of the things we would like to do is basically taking the in vivo data sets and comparing with the organoid data sets as you've shown. And here we will be having more formal links. We've started these links with the organoid network to try and kind of like have a more systematic comparison of the organs. That's one initiative that we've begun to do.
Thank you so much, Muz. We really have to go. They're telling me here we are running really late, but it was great to hear you. Really, really fantastic talk. Thank you.